Let's talk a little bit about enzyme inhibitors. So remember that we have substrates, which are the starting materials for enzymes. And the substrates are going to bind into the active site of the enzyme, and then the enzyme is going to change it to products. Now, we'll see that some inhibitors actually bind in that active site. We call these orthosteric inhibitors. These are often going to be competitive inhibitors, and you also see like covalent inhibitors or suicide or mechanism-based inhibitors that will bind in the active site. You can also have inhibitors that bind elsewhere on the enzyme, and we call these allosteric inhibitors. So with those terms underway, we can now move into talking about the different types of inhibitors based on how they do their inhibiting. So if you imagine a stick breaker and he's trying to break sticks, well, he's got to grab onto that stick. So enzyme plus substrate, you get this enzyme substrate complex. You've got to change that to the product and then let it go. Now, if he thinks it's grabbing a stick, but it's actually grabbing like a piece of metal, it's not going to be able to break it. Similarly, if the enzyme binds to a competitive inhibitor, it's going to bind to this kind of like fake substrate, and therefore it's not going to be able to make product. If it can't do this, basically what you're going to do is you're going to take this enzyme out of commission. But if you add enough of the real substrate, you'll be able to compete it out, because remember, this binding is going to be reversible. However, you can also have an irreversible inhibitor. An example of this would be like penicillin or beta-lactam antibiotics that actually bind to the enzyme and actually kind of trick it into attacking it. And in this case, it's going to get permanently stuck. And so that would just kind of permanently incapacitate your enzyme. Whereas competitive inhibitors can be competed out. So a purely competitive inhibitor is just going to bind to that enzyme alone. It can't bind it when the enzyme is bound to substrate, and it can't bind it when the enzyme is bound to product. Basically, it's binding in that same place, so the enzyme needs to be alone. And the competitive inhibitors are going to bind to the enzyme like this. And although I'm talking about substrate, typically if you want a really strong competitor, you're going to want to mimic the transition state. So remember the transition state, that's where the enzyme kind of hugs the substrate the most. That's that point at which you are on the cusp of becoming product, that really, really awkward spot when that stick is right about to break. And that's where the enzyme binds like tightest, because that's where you kind of need to lower the activation barrier by kind of providing that binding energy through all those interactions. So that transition state is what you're going to want your inhibitor to mimic in order to kind of best bind the enzyme and keep it from working. Those were orthosteric inhibitors, but we also have allosteric inhibitors where they're binding somewhere else on the enzyme. So you can imagine that the stick breaker, instead of picking up a fake stick, so it can still pick up the stick, but now even if it picks up the stick, it's got its like fingers tied together or something. It's somehow incapacitated. It can't do its job. It can't actually convert that to product. There are a couple of different potential scenarios. One is that the competitor can bind to the enzyme when it's either alone or when it's substrate bound. So we call this a non-competitive inhibitor. It's like, I'm not competing with you. I'm binding somewhere else. I don't care what you do. You do you. We're good. I'm just going to bind regardless. So this is going to be able to bind to the E or the ES. In a purely non-competitive inhibitor, you're going to have equal affinity for the enzyme in both the unbound and the substrate bound form. That would be purely non-competitive. However, we also see examples where we have mixed affinities. So maybe it'll bind to either, but it prefers if the enzyme's alone, or prefers that the enzyme's bound to substrate, because maybe when the enzyme binds to the substrate, it kind of undergoes a little shape change. Remember, we have that like induced fit and things with enzymes. So it's not that lock and key, but rather it's that induced fit where you get those initial kind of weaker bindings, and then it kind of molds around the substrate and forces it towards that transition state. And then in the transition state, when the substrate's the most awkward, the protein is hugging it the most, and then you go to your product. In the case of a non-competitive inhibitor, it's going to be able to bind to the enzyme in either of these. And also in the case of mixed inhibition, you're going to be able to bind to either of these sites. But in the case of mixed inhibition, it's going to prefer one of those sites a little bit more. This is going to have implications for what types of evidence it leaves in the inhibition. an uncompetitive inhibitor. Uncompetitive inhibitor is going to only bind to the enzyme when it's bound to the substrate. So non-competitive, remember, it's like, I don't care what you do you. Enzyme, then uncompetitive is now it's saying, I'm not competing with you for that enzyme. I'm going to be binding to the enzyme substrate complex. So this is basically, if your enzyme substrate, what can happen is either you get one of those shape changes, or you can get something cool like the binding, like when you have the substrate bound, it's going to kind of form a binding pocket. So this was actually what I was working on in my postdoc. So basically after I graduated and before I started teaching, I did a year in Sunita Fujimori's lab at UCSF doing some more research. I was studying antibiotics that target bacterial ribosomes, but that they can also target mitochondrial ribosomes, like off-target activity, because, well, basically our mitochondria come from a really ancient cell swallowing a bacterium or whatever. And so our mitochondria, they have their own ribosomes, and they're going to be similar to the bacterial ribosomes. And both the mitochondrial and the bacterial ribosomes have this cool thing where with some antibiotics, so like chloramphenicol and oxazolidinones, like linezolid, when a certain sequence is being translated, it makes like for a stable binding site for the antibiotic to bind and prevent translation. But only when specific amino acids are there. And so you get this like context dependent inhibition. And what happens is that as the nascent chain, so as the peptide, the protein's being made, it's kind of like winding out through the tunnel, the chimney of the ribosome, you might think about it. And when there's a specific, only when there's a specific amino acid or like characteristics amino acids, there are a couple that'll work. Basically, when it's in the kind of tunnel where you need to actually add the next peptide on, the next amino acid on, form that peptide bond. If the antibiotic binds there stably, then you can't do that. And the antibiotic only binds there stably in the case when they have a certain amino acid in the chain. And so the amino acid is actually able to stabilize the binding of the antibiotic. So this is like the case of uncompetitive inhibition, because when it's translating, when it's making the substrate for the enzyme to use, well, that's when it's going to make it so that the antibiotic is actually going to bind stably. And so you can read about it more in the preprint, as well as more work from the Fujimori lab. I'm also not going to get more into here. There's this thing called reversible covalent inhibition. We're going to basically, typically you just think about covalent inhibitors as being irreversible. So typically those covalent inhibitors are binding to that active site, doing one of those like suicide or mechanism-based inhibitions, where basically they're going to be they form bonds at that like active site, but they're typically to like a sulfur or something, and so they're going to be reversible, but they are covalent, so they're stronger than the competitive, but they can be overcome. And so this is a way that you can kind of like limit off-target activity, but not going to go into that here. If anyone's interested, I have a video on that into this. But for now, we're just going to focus on kind of these simpler cases. So we have these 4 cases of types of inhibition we'll talk about: competitive, irreversible, non-competitive, and uncompetitive. And remember that mixed is just a type of non-competitive where you have unequal affinities for your enzyme and your enzyme substrate complex. But they're irreversible are covalent inhibitors that are binding to the active site and basically keeping the enzyme from doing its thing. Typically, you're going to see this with an enzyme that has a covalent catalysis mechanism. As for what it's going to do with the kinetics, we're not going to bother with that. So you're going to have a decrease in activity of your enzyme. The effects with KM can be a little strange to think about because it gets all really technical and weird. And typically when you're dealing with these curves and stuff, and you're talking about the effects on KM and Vmax, you're typically dealing with these with reversible inhibitors, and that's going to be our focus. However, we'll look at some irreversible inhibitors and kind of how they can hijack enzyme mechanisms. An example of this is going to be our beta and lactam antibiotics. So the bacteria basically targets this transpeptidase that the bacteria uses to make its like sturdy cell walls. And then the ampicillin looks like kind of what the substrate for that transpeptidase. So the bacterial enzyme attacks it, but then it gets stuck and the cells can't make strong cell walls, and then they burst and die. Serine proteases, we can also use covalent inhibitors of them. And so basically what's going to happen is that these enzymes go and they think that this is a peptide, but really it's your inhibitor. And so they get stuck on it. permanently. Okay, now let's talk about our reversible inhibitors. So when we talk about our reversible inhibitors, remember we can have our orthosteric and our allosteric. Our orthosteric inhibitors, these are going to be binding in the active site, and these are typically going to be our competitive inhibitors. Whereas our other types of inhibitors are going to bind in an allosteric site. So remember that if you're able to bind to the enzyme when it's bound to the substrate, you've got to be binding somewhere other than just the active site, other than just like purely where the substrate is going to bind. So if you think about what these might look like, well, a competitive inhibitor, one of those orthosteric inhibitors, is going to look like your substrate or your transition state. Whereas one of those other forms of inhibitors, those allosteric inhibitors, it can look like pretty much anything. You can't really just predict what it'll look like because they can bind to various places, and you never know like where binding at one thing will influence the shape of another site and things like this. So let's now look at how these influence our kinetics. So an important thing to keep in mind is that in the presence of an inhibitor, like the inherent goodness of that enzyme isn't going to change. It still actually has those same like kinetic constants. So Km isn't going to change, like your actual affinity for the substrate isn't going to change, but it might look like it's changing. And so we can say like a Km parent might change. Similarly, your kcat, so your actual like ability to turn over the substrate into product, if there weren't any like inhibitor present, your ability to transform that substrate to product would not be affected. So your kcat, like your actual kcat or your actual Vmax isn't going to change, because remember the Vmax is just the kcat times the enzyme concentration. But what can happen is that inhibitors can make it seem like you have less of the enzyme, and therefore it's going to be like you're multiplying that kcat by less of a smaller number, and therefore your Vmax is going to be smaller, and your parent kcat is going to be smaller, and that sort of thing. But so basically what we measure might change, and so we call these like Km apparent and Vmax apparent. But what's actually like at the enzyme level, if the inhibitor were gone, the enzyme is still like okay, except for a covalent inhibitor, but the apparent goodness of the enzyme isn't changing, is the key point. So first, let's just review our Michaelis-Menten kinetics. So we basically have the situation where the enzyme binds to the substrate, and then it can bind and unbind and bind and unbind and bind and unbind, and those are given with rate constants of K on and K off. Once it's bound, you have this enzyme substrate complex that can then go and get catalyzed, converted that substrate into product, and this is given by our kcat. So the kcat is going to be the rate constant for that turnover, and we call this kcat, therefore, the turnover number. So that's telling you about like a single enzyme doing its best. How fast can it go? Km, this is going to take into account basically how much of this enzyme substrate complex we have, which is going to depend on how much you like the substrate, so how fast, how tightly you bind, as well as also a contribution of the kcat, because well, how fast you turn into product, that's also or how well you turn into product, that's also going to influence how much ES you have. So when we think about the amount of enzyme substrate we have, well, this is going to be like in roughly a measure of infinity, we have this Km, because the larger the concentration of substrate you need in order to get the same amount of basically productive product form is going to tell you that like, well, if you needed more substrate, you're not binding it as well probably. And so we say that the Km is a measure of binding affinity, where the lower the Km, the higher the affinity. And we can figure this all out experimentally by measuring the amount of the rate at which product is formed over time in different substrate concentrations, taking the slope of the linear portion, this V naught, and then plotting those out and finding the concentration of substrate at which we got our maximum velocity and our half maximum velocity. We can get the kcat from dividing the maximum velocity by our enzyme concentration, and we can get the Km by taking the concentration of the substrate that got us to half of that value. So this is going to be the basics of Michaelis-Menten kinetics. Now let's go and look at how these kinetics are going to be influenced by our inhibitors. Our competitive inhibitor. So our competitive inhibitor is competing with the enzyme for that active site. It can only bind the enzyme when it's alone, because, well, if the substrate's there, there's no room. So it's going to bind to the enzyme site. It usually is going to mimic the substrate and bind in the same place. Now, the enzyme, it has a choice. Is it going to bind the inhibitor or is it going to bind the substrate? Well, it's going to depend on its relative affinities for the two and the concentrations of each of them. If you don't have any inhibitor present, well, then that would be your normal Km. But what if you start introducing inhibitor? Well, now you're going to have more of your actual substrate in order to get to your half maximum velocity. And so it's going to look like you have an increased apparent Km. It's going to look like you have a worse affinity because it's going to take you more of the real substrate in order to get to that maximum velocity. But that maximum velocity itself is going to be the same because, well, if you add enough of the substrate, you're basically diluting out the inhibitor to such a degree that as long as the inhibitor is not permanently stuck on there, if it just like happens because to unbind, well, now the first thing it's going to pick up is going to be a substrate. It's more likely to pick up a substrate than it would be to pick up an inhibitor. So remember that you have this binding and this unbinding, this binding and this unbinding of the inhibitor, not just of the enzyme and the substrate. And so you have to think about, okay, well, if the enzyme were to drop the inhibitor, would it be more likely to find another copy of the inhibitor or a copy of the substrate? If it's more likely to find a copy of the substrate, and if it has a higher affinity for the substrate and things like this, well, then you're more likely to find it bound to the substrate than bound to the inhibitor. And so that's why you can compete these out and reach the same Vmax. However, the amount of the substrate that's going to take you to get to that Vmax into the half Vmax, that's going to increase. So remember that it's not the actual Km that's going to be changing, but the apparent Km is going to be changing because it's basically the real substrate is being diluted out. But we can dilute the fake substrate out as well by adding more of the real substrate. Non-competitive inhibitor. So here, it's like you have less enzyme. And so if we think about why this is, well, it's because the inhibitor is binding it and keeping it from doing what it's doing. And the way that we measure how much enzyme we have, well, I mean, like the way that we measure enzyme activity or whatever, we're measuring its activity. We're measuring the formation of product. And if we're saying that the enzyme can't do that, it can't form product, then it's going to look like there's not as much enzyme there. And so remember that the non-competitive inhibitor, we're binding to the enzyme at an allosteric site, so someplace other than the active site, and we're somehow incapacitating the enzyme, keeping it from doing its job, keeping it from converting the substrate into the product. So remember that purely non-competitive inhibitor is going to be able to bind to both the enzyme alone and to the enzyme substrate complex with equal affinities. So it doesn't care. It's not competitive with that enzyme for the substrate. It's not competing with that substrate. It's saying you do you, I'll do me. I'm going to bind my enzyme, the enzyme over here. Go about your business. And so if it's doing this, well, what's that going to do? It's going to make it seem like we have less of our enzyme. If we have less of our enzyme, so remember our Vmax, that was going to come from our kcat times our enzyme concentration. If we're saying it's like we have less of our enzyme, well, then it's going to be like we have less, we're going to get a lower Vmax. So we're going to get a decrease in our apparent Vmax. What about our Km? So remember, if we can simplistically think about Km as a measure of binding affinity. If this non-competitive inhibitor is not competing, well, then it's not going to change the affinity of the enzyme for the substrate. And so the apparent Km is not going to change. And so that's why we have, in this case of a purely non-competitive inhibitor, we have no change in the apparent Km, but we do have a change in the Vmax, the apparent Vmax, because the competitor binds in a different location. It's not competing, but it is keeping the enzyme from doing its job if the enzyme does bind to that substrate. Often what we're going to see, though, is mixed inhibition, where there are different affinities for the E and the ES. So non-competitive is just a special case of mixed inhibition. With mixed inhibition, you're still going to get that lower Vmax, but your Km can either go up or it can go down. It's a little confusing, but hopefully it'll make a little more sense once we talk about the uncompetitive inhibitors. But basically, you can think about, if it's preferring one form over the other, like if it's preferring the enzyme alone form, well, then it would be kind of like drawing this and pulling your reaction this way and making it seem like your enzyme had worse binding. So that would be kind of like your competitive inhibitor, which was going to increase your apparent Km. Well, if it was binding to this enzyme substrate complex better, well, this would actually make it look like it had tighter binding, because if you think about Le Chatelier's, you're pulling things this way, you're going to have increased binding. It'll look like you have increased binding affinity. You're going to decrease your apparent Km, that is, increase your apparent affinity. So hopefully it'll make a little more sense since we talk about uncompetitive inhibition. So remember your uncompetitive inhibitors, that's where you're binding to the enzyme substrate complex, but not to the enzyme alone. And you're preventing it from being, the substrate from being converted into product. So when the enzyme binds to the substrate, now you might have like a little site open up or something. That's going to allow the inhibitor to bind. And once the inhibitor binds, it's going to be incapacitating it. So if you think about our types of inhibitors, if you think about kind of the competitive inhibitor, that's going to be like your fake stick. If you think about a mixed inhibitor or a non-competitive inhibitor, that can be kind of like you're grabbing onto the stick snapper's wrist, but just kind of in either way. And so the stick snapper can still grab onto a stick, but it can't do that kind of like movement of its wrists in order to snap the stick. What you're saying when you're saying it'll only bind to the enzyme substrate complex, like the case of uncompetitive inhibitor, is maybe only like when that muscle group or whatever, when you're in that form that's bound to the substrate, only then are you going to grab onto their hands and keep them from doing what they want to do. So because you're keeping the enzyme from doing what the enzyme does, you're going to decrease your apparent Vmax. However, you're going to get kind of a weird result with your Km. Because what you're going to be doing is you're going to be drawing the enzyme substrate complex like to the right. So you're going to be making it like good old Le Chatelier's, you're drawing things this way. You're going to be increasing it's going to look like you're increasing the affinity of the enzyme for the substrate because you're going to be kind of like pulling it out here. And if we're saying that you're increasing the apparent affinity, what that means is that you're going to be decreasing your apparent Km. So it's going to take less of the same amount of substrate in order to get to your half maximal velocity, but the half maximal velocity is going to decrease. And so your enzyme and substrate complex becomes stabilized, but they're kind of useless. So to summarize, we have this enzyme bind to the substrate. We get this enzyme substrate complex that gets converted into the enzyme and product. So remember, we kind of lump together the forming the product and releasing the product, and we say that that's kind of like our kcat. And then the binding to the substrate, that's kind of like our Km, but the Km also takes into account this part. So it's not as simple as saying binding affinity, but it's often just considered a measure of binding affinity. So similar to kd, and that the lower the better, but there is that extra complicating factor. And so it's not completely independent. It's not completely affinity, but we can approximate affinity. And now let's say we have an inhibitor. We have a competitive inhibitor. It's going to bind to the enzyme and alone. It's going to prevent the enzyme from getting to the enzyme substrate complex. Well, what this is going to do is if the enzyme can't get to that, you're going to need more of it. You can compete it out because the enzyme isn't prevented from doing its business. If it actually binds the substrate, it can still stop it. But it's like the stick snapper needs to find more it keeps finding these fake sticks. So we can compete it out. If we add enough of the real sticks, we can kind of make it so that when the stick snapper drops that stick, it can find another one, it can pick it up and keep snapping. And so because remember on our Michaelis-Menten curve, we have like substrate on the x-axis. If we have enough of that substrate, we can finally compete out the inhibitor. And so we're going to get this to the same Vmax, but we will take us more of our substrate to get there. So it's like we have a lower affinity. That is, we have an increased apparent Km. So a competitive inhibitor, we can compete it out with substrate. With our other types of inhibitors, we cannot compete them out with substrate. And if we can't compete them out with substrate, that basically means we're going to take some of our enzyme out of commission. And if we take some of our enzyme out of commission, well, then we're going to decrease our apparent Vmax. What might change is our Km. And it could go up or down depending on our type of inhibitor. If we have a non-competitive inhibitor, what this is saying is if it's purely non-competitive, it's going to bind to the enzyme alone or to the enzyme substrate complex. Because it can bind each of these equally well, then it's not going to change the affinity or the apparent affinity of the enzyme for the substrate. You do you, I'll do me, I'm binding over here, I don't care what you're doing. So that's going to be your non-competitive inhibitor. So that's not going to change your apparent Km, but you will have that decreased Vmax. With mixed inhibition, you're going to have non-equal affinities to the enzyme alone and the enzyme substrate complex. Then things can get a little complicated. And so the Km could go up or down, depending on whether it acts more competitive or more uncompetitive. So with an uncompetitive inhibitor, remember here, we're binding only to the enzyme once it's bound to the substrate. And once the enzyme is bound to the substrate, it's going to, basically the inhibitor binds and prevents the substrate from being converted into product. What this is going to do is it's actually going to pull the reaction this way. You're stabilizing that enzyme substrate bound form. You're pulling the reaction this way. You're going to make it seem like it has a higher affinity. You're going to decrease the apparent Km. But you're still going to get that decrease in your Vmax that you measure. So with these non-competitive and uncompetitive inhibitors, you cannot compete them out, but you can dilute them out. So when we say compete them out, that's like we're adding more substrate and we can get to the same Vmax. And the only time we can do that is if we have a competitive inhibitor. But we can dilute them out, where basically if we're diluting them out, that basically means we just kind of wash them out. So that if the enzyme can't find an inhibitor to bind, well, then it's not going to be bound. You can't compete these out because, well, they're not competing for the same site. So even if you add a bunch more substrate, the substrate binding wasn't the problem. The substrate has plenty of sites to bind, and the inhibitor is not preventing that. If you want to keep the enzyme from binding the inhibitor, you have to make it so that there's like less of the inhibitor. Make the effective running out of inhibitor by diluting it out, so that when the enzyme goes and because remember the inhibitor is binding and unbinding, if it unbinds it but it can't find another inhibitor to bind, well then that is going to be able to increase the enzyme. Like the enzyme is going to get kind of like back to normal, because that's non-covalent binding, as opposed to our like kind of suicide binders, our mechanism-based binders, our covalent inhibitors. With the competitive inhibitor, here you can dilute it out or you can compete it out. And the reason why you can compete it out is because if you have real sticks that it can pick up, the only thing that was keeping it from doing its making product was that it couldn't find the sticks, or that like it was finding the fake sticks instead of the real sticks. And so if we make it so that there's more real sticks than fake sticks, a lot more real sticks than fake sticks, then we can compete it out and get to that same parent Vmax. So to summarize, competitive is going to bind to the enzyme alone. It's not going to change our Vmax apparent, but it's going to increase our Km apparent. Our non-competitive, this is going to bind to E and ES with equal affinity. It's going to take that enzyme out of commission. That's going to decrease our Vmax apparent. But if you're binding to E and ES with equal affinity, that's not going to change your Km apparent. Uncompetitive. Here you're binding to ES and only ES. That's going to decrease your Vmax apparent. Remember, you're taking the enzyme out of commission. And then that's going to do, though, is it's going to basically stabilize that substrate bound form. It's going to pull the reaction towards the enzyme substrate complex. That's going to decrease your apparent Km, make it look like you have a higher affinity. Mixed inhibition. Here you're binding to E and to ES, but with different affinities. Once again, you're taking the enzyme out of commission, decreasing your Vmax apparent. And but because you have different affinities, this may increase or decrease your Km apparent. If it acts more like a competitive inhibitor, binding to that enzyme, like the enzyme alone titer, well, that's going to increase your apparent Km, just like you had with your competitive inhibitor. But if it acts more like an uncompetitive inhibitor, well, then it's binding to the enzyme substrate complex more, and that's going to decrease your apparent Km. So you can't really tell, it's hard to tell based on a graph or something like that, whether something's mixed. Mixed is actually a very common form of inhibition. Now remember, all of these are changing what you're measuring, so they're changing the apparent things, but they're not changing the actual kinetic constants, the actual Km, the actual kcat. They're just changing kind of what you are measuring. And in the case of our competitive inhibitor, we can compete or dilute it out. And in the case of our non-competitive, uncompetitive, or mixed inhibitors, we can dilute them out, but not compete them out. And in the case of our covalent competitor, or like our irreversible inhibitors, we can't do any of those. The only way to kind of get activity again would be to add more enzyme. Often when you're talking about trying to figure out what type of inhibition thing is, you might use a Lineweaver-Burk plot. This makes it easier to kind of see the differences in these key values fairly quickly, but use caution with Lineweaver-Burk plots because of the whole deal where the points far from the origin, where you have a really low substrate concentration and your data is probably not that accurate anyway, are going to have a kind of like lever effect where they're going to pull the slope and greatly influence the values. But remember that with a Lineweaver-Burk plot, you have 1/s on your x-axis and 1/v on your y-axis instead of s/v. So this is what's going to happen is then your -1/km is going to be your x-intercept, and 1/Vmax is going to be your y-intercept. So if your apparent Vmax gets smaller, what's going to happen is that this is going to get shifted up. And if your Km or your apparent Km gets bigger, what's going to happen is that this is going to get smaller. So if we have competitive inhibition, remember that's going to increase our Km. So in the presence of our inhibitor, if the Km was getting bigger, this is going to get smaller, it's going to get closer to the origin. And we're not having a difference in our Vmax, so we're going to have the same of y-intercept. In the case of uncompetitive, so remember that was going to decrease our Km, so that's going to shift our x and our y-intercept out here, and it's going to decrease your apparent Vmax, and so that's going to increase this intercept. And in the case of our non-competitive, which remember is a form of mixed inhibition, in the purely non-competitive form, you're going to have your Km unaffected, and so your x-intercept is going to be unchanged. But because your apparent Vmax is going to be reduced, what's going to happen is that, once again, you're going to see that this, with the inhibitor, you're going to be shifted up because that intercept is going to be the 1/Vmax. And if this is going to be smaller, this is going to be bigger. When it comes to measuring the kind of effectiveness of an inhibitor, we often talk about a couple terms, Ki and IC50. So basically your Ki, that's like your binding constant or your dissociation constant for your inhibitor, kind of like how we had a Kd when we did binding thermodynamics, and kind of similar to Km, how that was measuring affinity for the substrate. The Ki is going to be measuring affinity for the inhibitor. So you can imagine that the more effective, the stronger it binds, the inhibitor binds, then the lower the Ki, and thus kind of like the less of the inhibitor you need in order to have a response. And we can talk in terms of the response as well. So we have this term called the IC50, and the IC50 is going to be the concentration of the inhibitor at which you kind of get that half maximal effect. So maybe you reduce the enzyme's activity by half, or you might see this term used in other contexts where you kind of reduce binding by half and things like this. So to summarize, competitive, you're competing for the substrate, but can fall out and let substrate bind. Non-competitive, you bind to E or ES, and uncompetitive, you bind to ES, but not E. With a competitive inhibitor, you reach the same Vmax, because if you add enough of the legit substrate to outcompete all the fakes, the enzymes sound act normal. But you have an increased apparent Km because substrate gets diluted by the competitor and becomes harder to find. But we can basically switch the scenario and compete out the inhibitor to make the inhibitor harder to find. In the case of our non-competitive, well, we're going to have a decrease in our Vmax because not all the substrate-bound enzyme is actually able to do its job. But we're not going to have a change in our parent Km because the competitor binds to a different location, doesn't affect the enzyme substrate binding, isn't competing. With our uncompetitive, well, here, not all the substrate-bound enzyme is actually able to do its job, so we're going to get a decrease in our Vmax. But because the enzyme substrate complexes become like stabilized, even though they're useless, you're going to get a decreased apparent Km. And then remember, we have our irreversible covalent inhibitors, where they're going to bind and basically trick the enzyme into thinking that they're substrate and attacking it. And so this is typically when we have one of those mechanisms where the enzyme is actually forming a covalent link with so it has a covalent catalysis mechanism, and therefore it normally forms like a temporary link with the substrate. But in this case, it's going to form a permanent link with the inhibitor, because basically the inhibitor is going to be a worse leaving group, and so it's going to get stuck on there, preventing the enzyme from regenerating. And a classic example of that is going to be like your ampicillin and your other beta-lactam antibiotics.